So a few things that I plan on covering today. We have in genetics, we have left mutations and the Ames test, horizontal gene transfer, and regulation of gene expression, which I will merge with enzymes, kind of do a big chunk of it. So let's start with the mutations. We've already mentioned before that I think we went over different types of mutations. Yeah, so we're going to talk about mutations in the context of mutagens. So mutagens can be divided into two categories, as you remember: chemical mutagens and radiation. We're going to start with the radiation. And there are two types of radiation that can cause mutations. One is non-ionizing. It does not result in the formation of ions. Basically, non-ionizing radiation is the UV light. Okay? The effects of non-ionizing radiation on the DNA usually manifest in the form of pyrimidine dimers. Often it is exemplified, and even like replace the whole idea of pyrimidine dimers, is thymine dimers. So thymine is one of two pyrimidine nucleotides that are found in the DNA, and this is how it looks. Look, you have two DNA strands, right? Antiparallel is all as usual, and you have two thymine nucleotides next to each other. When they are exposed to UV light, the bond between them forms. That formation of a bond results in the distortion of the DNA strands and eventually may lead to either point or frame shift mutations. We good so far? Okay, next type of radiation that is worth mentioning is ionizing radiation. Out of all the types, there are two types that are used in microbiology: X-rays and gamma rays. That symbol here is the Greek letter for gamma. Okay. So ionizing radiation causes single or double strand breaks Obviously, single strand break isn't such a big deal because you have a backup in the form of a second strand, but double strand break is kind of a catastrophe. This catastrophe is being fixed by various repair systems, but the problem is that repair often results in mutations. And these mutations can be either point or frame shift. Now what about chemical mutagens? So chemical mutagens can be divided broadly into three groups. The first group being nucleotide analogs. So imagine that during the DNA synthesis, the nucleotide that is inserted is not a proper one, neither A, G, T, or C. So that results usually in the point mutations. And point mutation in our case will be the replacement of the pair. So for instance, at certain location in the DNA, you had G-C pair, and it is replaced by A-T pair. Okay, does that make sense so far? Okay. Another really interesting agent, chemical agent that causes point mutations are, they sometimes called nucleotide modifying agents, but we're going to talk about nitrites or nitrous acid. So this is basically the anion, NO2 minus. Nitrites are used in the food production to preserve freshness and color of processed meats, but turns out that nitrites can cause point mutations of the same sort. And if they can cause point mutations, I mean they can preserve those meats from bacteria, but the problem is they also can affect human cells, and currently nitrites are being kind of pushed out of the food industry because they can be potential carcinogens. And finally, the third chemical class of chemicals, so-called intercalating agents. So these are really cool chemicals that do this. Molecule of intercalating agent, look here, it gets inserted between the pairs of nucleotides, like my hand gets inserted between, these two pairs. Does that make sense? So this insertion changes the shape, the double helix, and even changes the shape of a single strand. So this results, write it down, between the pairs, and that results in a frameshift mutations. Any questions so far before we are moving on? We good? Okay, so we have a couple more items to talk about in the context of mutations. First, how can we fix them? In order to illustrate this idea, I'm going to use two examples of repair systems. Those are just two examples that you need to have an idea about, and they will be relevant for the thymine dimers. So imagine you have DNA that was exposed to UV light, and thymine dimers are formed in that DNA. So how can it be fixed? One way to do that, if the light is still there, okay, so one outcome will be light is still present. So in this case, your thymine dimer so you have DNA, you have two thymines next to each other, and they are connected by a chemical bond. So this thymine dimer is exposed to the enzyme called photolyase. Okay? And photolyase does an incredibly clever and, quite frankly, simple thing. It simply breaks the bond, and that is all. So you go back to square one, you go back to normal. Okay, thymines are not connected to each other anymore. Does that make sense? Now, second option, when there's no light, okay, there's no light, you go through the following steps. So this is your DNA, okay, double-stranded DNA, and I'm going to use an asterisk to mark thymine dimer, so I don't have to draw that all the time. So this asterisk represents your thymine dimer. So the first step that happens here is the enzyme called endonuclease introduces the brakes into the strand with the damage. Okay? After that, your old pal helicase moves the fragment with a mutant out. Okay? Does that make sense? Here. It just takes it out. Now you have a gap. So all you need to do is to fill up that gap. So the enzyme called DNA polymerase 1, which we encountered already when we were talking about the replication process, it fills the gap. However, this fragment that fills the gap needs to be attached to the original strand, and this is what's done by the enzyme called ligase, also known to you from our conversation. So ligase stitches them back, and just show by these dots, I show the places for stitching. Okay, so now everything is repaired. Does that make sense? This is not by any means the only mechanisms for repair. There are more, but those are kind of basic ones: photolyase and excision repair. So this is called excision repair. Any questions so far? Good? Okay. Now, really big concept, methodological concept that I want to introduce to you will be the Ames test. So what is the goal of Ames test? Imagine that you have a drug, or any chemical that you want to use in humans. Of course, you don't want that chemical or that drug to be carcinogenic. Okay? If I ask you how you test for ability to cause tumors, probably the first answer will be, I'm going to inject it in mice. Well, great, but it will be really hard to get a medical history from them, because last time I checked, they can't speak. Also, if they form tumors, the question is where? Where is the tumor? You can do a really expensive MRI of mice, that does exist, but if a tumor is rather small, will you be able to find it? Okay, so it gets really, really complicated with animals. You need something easier and something more efficient to triage chemicals by their ability to cause mutations. And this is the goal of Ames test, to check if a chemical can cause mutations. Am I clear? So in order for, I don't want to introduce some new species, whatever, so I'm going to use basically the same example that's in the lecture notes, but I'm going to go through that step by step, okay? And I'm going to walk you through this. So the test starts with the petri dish on which you have salmonella. It doesn't have to be salmonella, but let's use salmonella just for the example. So this is a garden variety salmonella species that grow pretty well on a defined medium. Okay, so those blue are colonies of salmonella. So I'm going to make a couple of statements. They're not complicated statements. Don't read anything more than they say into those statements, okay? So statement number one, really simple. This medium contains amino acid histidine, okay? It's not like some epiphany. So we're going to say that this medium is histidine positive. Just make this little notion that there is histidine in that medium. And the second statement that I'm going to make is that this particular species of salmonella can make amino acid histidine on their own. So they are histidine plus. Okay, I'm describing you the phenotype. Okay, we good? Then we do what's called a replica plating. We take a piece of fabric, a circular piece of fabric, okay, a round piece of fabric. We press it against this agar plate. And we basically do a Xerox copy of that plate on two other agar plates, okay, to replicate the arrangement of the colonies. So these two plates now are slightly different. So this plate on the left still contains amino acid histidine. The plate that is on the right doesn't. So agar here in this plate doesn't contain an amino acid histidine. Okay, good? So here on this plate, you're just going to get the exact copy of the colonies that you had on the first plate. Are we clear? Now on the second plate. You look at it and you notice that there are only 4 colonies. Okay? Which makes, look, this colony right here somewhat unique. So what is the uniqueness of that colony? I think about it in the terms of, nutrients that a microorganism needs to live. So these microorganisms in this colony, they can grow when histidine is present in the medium. But when histidine is absent, they can't grow. What does it tell us? It tells us that while these colonies, these microbes, can make their own, this one cannot. Does it make sense? I'm not going to move on until, so please don't treat it as I'll just tell it makes sense so he can just shut the fuck up and move on. Okay, please, yeah.
If they're just making copies of the plate, how does the one on the right become negative?
Say it again?
So you said it's like making Xerox copies basically of the plate. So how does the one on the right become negative?
Good question. So I didn't explain it well, I think. When we do replica plating, we copy the arrangement of colonies. The medium in this plate, the medium, the agar, is slightly different. And its difference is that it does not have histidine in it. Okay? So far, are you following me? So we've got this colony, which is also salmonella, but this salmonella can't make histidine on their own. So we're going to call it histidine minus. This is called auxotrophic mutant or auxotroph. Auxotroph is an organism that cannot survive without a particular nutrient in the environment. Humans are ultimate auxotrophs. We can't survive without essential amino acids. Questions? Okay. Question? Go ahead.
So does the agar on the left have something different from the one on the right?
Yes. Agar on the left has amino acid histidine in it. Agar on the right doesn't.
And then the first one has it too.
Yes. And the first one has it, yes. Does that make sense? Moving on?
Yes. Can you say again what the Ames test is?
This is the whole thing. This is the whole thing.
I know, but before you started, can you just say again what you explained it as?
We trying to identify if a certain chemical causes mutations.
Okay.
We aren't there yet. You will see, when we get to the chemical moment, it will be loud and clear.
Okay.
Because now what I want to go back to is like a very first conversation in this class when we mentioned that phenotype of an organism is defined by the gene expression. Does that make sense? That pretty typical statement. Now, inability to make histidine is definitely a phenotype. Does that make sense? So, histidine is very likely, not very likely, but histidine is generated by certain enzyme or enzymes. For the simplicity, let's say, microorganism like this salmonella has DNA. Within that DNA, there's a gene that is responsible for synthesis of histidine. Does that make sense? And look, in this blue salmonella, let's call it a blue salmonella. In blue salmonella, that gene responsible for histidine synthesis is absolutely functional. Does that make sense? It works. This gene is good, there are no mutations. When it is expressed, it produces proper enzyme that synthesizes histidine. Is that clear? Okay, let's talk about this salmonella then. I'm going to ask you a series of questions. So again, we have DNA. Does this salmonella have a gene for histidine? Yes. Thank you. Very good answer. Yes, it does. Is it functional? No, it is not. Does that make sense? So I'm going to put, again, an asterisk here. Are you following me? I used an example, a couple of examples yesterday because for some reason humans just are easier to understand. People who are lactose intolerant can't break down the sugar, lactose, right? Because they do not produce the enzyme called lactase. Do they have a gene for lactase? Yes, they do. Is it working? No, it does not work. Does that make sense? So, say I am not lactose intolerant, I'm fine, I have that gene. A buddy of mine is lactose intolerant. He does have that gene, but his gene produces dysfunctional protein or just not good. My gene produces functional protein, okay? So, transitioning from humans to bacteria, both histidine-producing salmonella and histidine-not-producing salmonella have the gene. But in this one, when this gene is being expressed, the protein is okay. When this gene is being expressed, the protein is dysfunctional. Why? There is a mutation in that gene. Some absolutely random mutation. Does it make sense? So basically what we did, we identified a very precise phenotype that is associated with a mutation. No chemical has been introduced yet. Up to this point, are you on board with me? Now, a really important statement, which I ask, like, you either, it's a correct statement, but like, agree with me or not, but the statement is this. If I will expose this DNA, this salmonella, to some kind of a mutagen, there is a non-zero chance that in some cells, this mutation will be reverted back to normal. Does that make sense?
Can you say it one more time?
So, if I expose this mutant salmonella to a mutagen that I'm testing, then there is a chance that this mutagen will correct the original mutation. Does that make sense? No? Does it? Okay. So this is what we do. We essentially expose it to a chemical that we are testing. We don't know if it's a mutagen. So if it is, and this mutation, so basically think of it this way. The original was GC pair, okay? In the mutant, it has been changed to AT pair. If this site gets mutated again, it becomes GC again. Make sense, right? So if this is a mutagen, what we're going to get out of that stuff is, again, salmonella that can produce histidine. Is that clear? Good? That makes sense. Now, if we expose salmonella to, for simplicity, to water, is water going to cause the mutation here, the reversion? No, it's not. So we're going to get the same non-histidine producing salmonella. Now, next step that we need to check. We need to plate the post-exposure bacteria on agar. Which agar should we use? The one with histidine or without? Yes. So, you're the first to answer. Hold on, I'll just repeat. So, after the exposure, let's focus on this one. After the exposure, we need to plate the bacteria after they were sitting in the presence of that chemical. Which agar should we use? With histidine or without? Without. Because we need to check if whatever comes out of the exposure can start producing histidine. Does that make sense? This medium doesn't allow us to distinguish between the two. Medium without histidine does. So, if our chemical is mutagenic, then the salmonella will be growing on histidine negative agar. And if our our chemical is not mutagenic here, then this red salmonella won't be able to grow. Does it make sense? So basically, kind of going back to the whole goal of the Ames test. Look, you apply your chemical and then you plate whatever you get after the exposure. If you get the growth, you're dealing with the mutagen. If you don't get the growth, your chemical is safe. Does that make sense? Good? So in other words, if chemical is mutagenic, then you get the growth on histidine negative medium. If chemical is non-mutagenic, you don't. Okay? Before we wrap Ames test, questions about it? Good?
So
Okay.
all that's done just to make sure that it's safe enough to put in a human.
Well, whatever you want to put it in, but it's cheap, fast, and it gives you, like, you can triage chemicals right away. What I'm saying is, if you test a new drug and you get this, it's gone. If you test a new drug and you get this, maybe it's toxic, maybe it does some other terrible shit, but at least it's not mutagenic, and you can kind of move on to the next testing step. Make sense? Good? Okay,